Okay, in this part, we're going to talk about mutations. So, mutations are changes that can happen in the base sequence of the DNA. Sometimes these changes at the DNA level can cause changes at the protein level, but sometimes the changes at the DNA level or the mutation at the DNA level cannot cause any changes at the protein level, that we call it actually silent mutations or neutral mutation. So there's different kinds of chemical and physical agents that can result in production of the mutation, and they call them mutagens. So the first type of mutation, they call it point mutations. This is when just one single base or nucleotide base pair changes in the DNA sequence. Sometimes just by changing one nucleotide, for example, instead of uh, think about if you look at this example, so this is our wild type DNA, so this is like a DNA that doesn't have any mutation. This is like a healthy person. Imagine like there is no mutation in that in the DNA of that person. So if one of the nucleotide changes, for example adenine changes to thymine in our DNA, so then when the DNA changes, messenger RNA gonna change, and then when the messenger RNA changes, the codons, if you remember every three nucleotides on messenger RNA, we call it codons. So then the codon is gonna change, and then amino acids gonna change. The first type of mutation that I call them like kind of good mutations, that the DNA changes, the messenger RNA gonna change, but if you remember, the codons are redundant, so even the codon is changed, you can see the changes in the codon, but that codon can still make the same amino acid, or can encode the same amino acid, so we call this silent mutation. So the changes at the DNA level cannot cause any changes at the protein level, so the amino acid gonna remain the same, so that's why they call it silent. It's like nothing happened. We have missense mutation. So this is when one nucleotide changes on the DNA that can cause a different amino acid in being replaced with the original amino acid. For example, instead of the this amino acid that you can see it in the wild type, because of the changes in the DNA and then change in the messenger RNA, so the codon before it was CCC, now it's ACC, so it's gonna encode a different kinds of amino acid. So we call this missense mutation. And then we have nonsense mutation. So this is when one nucleotide changes on your DNA, and then messenger RNA gonna change, but unfortunately the codon, because of the mutation that happened, is gonna cause like, or it's gonna make an early stop codon. If you remember, we talk about the stop codon, and in and it's going to result in the synthesis of a protein that are proteins that are shorter than the wild type, and typically they are not functional. So we call this nonsense mutation. So silent, missense, and nonsense, these are different types of point mutations that can happen. We have frame shift mutations. This is when one or two nucleotides inserted or deleted from our DNA that can cause changes at messenger RNA level, and then it can cause changes at the protein level. So, frame shift mutation caused by insertion or deletion of one or two nucleotides that are not, and you have to make sure that it's not a multiple of the three, and then it can cause shifting the frame of the translation, and it's going to change the polypeptide sequence, and then it can cause like proteins that are not functional, or they can have like even early stop codon that can cause like a nonsense mutation, and then it can cause like proteins that they cannot anymore do the job that they were doing previously in your body. So you can see in this picture, this is the wild type. So if you remove two nucleotides from your DNA. So we call this like frame shift mutation, so deletion of one or two nucleotide. So then if you remember, I told you every three nucleotides can make one codon. So when you delete two nucleotides, so it's gonna change the order of the codons. So before it was AGC, GUA. Now because you deleted those two nucleotides, so and every three nucleotides can make a codon, so then it's gonna completely shift and change the frame of translation. So now instead of AGC, GUA, CCC, UAC, you have AGC, GCC, CUA, CUU. So, and then based on that table that I've showed you for translation, when you have different frame of translation, you have different codons now due to the deletion of these two nucleotides, then you're gonna make completely different proteins. Sometimes it can cause even early stop codon. So if one of these codon be a stop codon due to the deletion or insertion, so it can cause like very short or not complete protein as well. There are different causes of mutations. Spontaneous mutation can happen, but it's very rare. So we have a lot of chemical mutagens that can cause the changes, like a point mutation or a frame shift mutation in the DNA. So usually these chemicals are carcinogens. For example, nitrous acid. So it can cause the change of adenine and thymine to guanine and cytosine. Or we have the base analogs that they have a similar structure as nitrogenous bases. They are a bit different than the nitrogenous bases, but they have a very similar structure that it can cause like mismatches in the DNA and then messenger RNA and in the protein as well. So, and then we have intercalating agents that they can cause like the insertion or the deletion of the nucleotides, and they can result in frame shift mutations. Also, if you remember, radiation can cause mutation too. Ultraviolet light, so these are like non-ionized radiation that can cause like the two thymine next together to make like bond with together, that they call it time they can cause dimer, or so these thymines, they call it thymine dimer, so attached together, and then it can cause the stop of the replication, transcription, and translation. And also ionized radiation, such as like x-rays or gamma rays, that are very strong and they can penetrate actually into the tissue and into the organism, so they can cause the formation of the ions and also free radicals, and then it can cause damages in the DNA. They can cause point mutation or substitution, or even they can cause the breakage in the sugar and phosphate backbone as well. So, but the good news is that we can repair the DNA. So there are some important enzymes such as DNA polymerase, that they have a very important characteristic that they call them, that we call it proofreading. So these enzymes, they actually can read and edit those mismatches, and they can replicate the correct nucleotide and replace those mismatches with the correct base pairs. And so they can do the proof, they can do proofread, they can proofread their own work, and we can repair the mismatches. Also sometimes, especially in bacteria, we can repair those thymine dimers, those thymine that they can make a bond, they can make bonds with together with exposure to the light, or with the nucleotide excisions. So we can correct those thymine dimers as well. So Ames test, this is a very common, and it's a very important test that they usually do it in the, especially in the pharmaceutical field, when they discover like a new medication or drug. So this is one of the tests that they do. So this is a technique that they use like bacteria for rapid and inexpensive screening carcinogenic potential chemicals. So what they're gonna do, so um, they're gonna culture the bacteria that exposed to that chemical or to that drug, and then they're gonna use a velvet, and this media, for example, has a specific amino acids such as histidine. Then they're gonna use a velvet, and then they're gonna press it on the bacteria colonies, so it's gonna print those pattern of the bacterial growth on the velvet. Then they're gonna have two kinds of media, one with the histidine and one without the histidine. So they're gonna transfer these bacterial colonies to these two types of the plates or media that they have, and after incubation, so they're gonna compare it and to see that which bacteria is missing in the culture that lack that amino acid. So if that colony is missing, so it means this bacteria is not able to make histidine or this amino acid to grow in the media without the histidine. So this bacteria is actually mutated, so it lost the capability of production of the amino acid that they need to survive, like histidine. So, and actually the loss of a function of the mutant, for example, loss of the function of production of histidine in salmonella, in this bacteria that we give you an example for the Ames test, they call it auxotrophs. So based on that pattern that they get in these two media, they can identify the ability of that drug or that medicine, that to see if it can cause mutation or it can induce the mutation in the, for example, cells or not. So this is like one of the primary techniques that they use to make sure that the drug is not carcinogenic or is not problematic for the health of the cells or the patient. So at the end of this chapter, we're going to talk about how prokaryotic cells, they can get genetic diversity. As you know, evolutionary diversity is a very important thing in the survival of the macroorganism. For eukaryotic cells, there is no problem because they have like a sexual fertilization, and they can cause like they have meiosis and all those processes that can cause the differences in eukaryotic cells. But prokaryotic cells, they just have asexual reproduction, so they're gonna do the binary fission and then they're gonna make like exactly the same bacteria or same genetic material as the original one. So how we can get to this diversity in prokaryotic cells? There are different techniques or different ways. One of the methods or one of the process that the bacteria follow in order to cause more diversity, we call it transformation. So bacteria, they have this capability that they can take the DNA that they have in their environment, so they can engulf the DNA or the fragment of the DNA or the plasmid in their environment, and then they can bring it into their cytoplasm, and then sometimes they can incorporate it with their genomic DNA as well, so it can give them like new characteristics that, for example, they didn't have it before the transformation. So if I just want to show you very simple, so imagine this is the bacteria, so this is a piece of the DNA in their environment. So this is like a DNA in their environment. So they easily can engulf this DNA and bring it to their cells, and then sometimes it can become even part of their genomic DNA as well, that you can see it here. And then imagine this DNA carried antibiotic resistance genes, or it can have like toxic production genes, so that it can give like a new characteristics to the bacteria. So we call this transformation. Then the next way, we call it transduction. So bacteriophages, that we talk about it in chapter 6 about when we talk about viruses. These are the viruses that they can infect the bacteria. So this is a bacteriophage, and they have this like interesting complex capsid, and this is like their DNA. So they can land on the bacterial cells, and then easily they can inject their genetic information so to the bacteria, so they can infect the bacteria. So this genetic information can get can become part of the genomic information of the bacteria, and it can be generalized or it can be specialized. So some of them, they can enter to a specific area of the chromosomal DNA, or sometimes they don't care, so they just see it, and they can incorporate with just part of the DNA of the bacteria. And this especially happens in the lysogenic viruses. So then imagine this virus has some specific genes for production of for giving some specific characteristics, for example production of toxins that actually happen in diphtheria bacteria. So with this they can get some new characteristics that they didn't have previously. So the transduction, so it happens with the help of, this is the genetic diversity that can happen in prokaryotic cells with the help of bacteriophages, that they are type of viruses. Then we have conjugation. So bacterial sex, so it's different than the fertilization that happened in the eukaryotic cells. But during conjugation, imagine we have two bacteria. If you remember pili or pilus, so these are like a small extension that you can find it in bacteria. So one of the bacteria has like a plasmid, so we call it F positive or donor cell, and the other bacteria doesn't have that plasmid, so it's going to be F negative or receptor. So with the help of the pilus, so these bacteria are going to connect to each other, and then the plasmid or this genetic information can be transferred, or a copy of it can be transferred from the donor cells to the recipient cells. So then this bacteria are going to have the same genetic information that the donor bacteria cells has it. So this is actually very important in the antibiotic resistance process. So imagine that one bacteria has a plasmid that carries the antibiotic resistant genes. Then through the conjugation, so it's gonna introduce this genetic information to them, to another bacteria. So then gradually the number of the bacteria that they are antibiotic resistance gonna increases, and then we have to find new antibiotics, or we have to use the combination of the antibiotics to attack these bacteria.